Okay. Okay. So, yeah, I'm Dr. Raghvendra Mall. I'm a senior computation scientist in the Department of Immunology at St. Jude Children's Research Hospital. Before talking about the challenge, I would like to say that having participated in multiple dream challenges and being top team in leaderboard but never winning, it is very gratifying that I am presenting finally a winning solution at and it's a dream come true to be presenting in the dream podium. So as highlighted yesterday also by the panelists, one of the panelists, we were actually, a dream actually gives you an opportunity for people from a very small country like Qatar, who basically made up this team, and predominantly are the people who made up this team, uh, along with collaborations with people in US and Italy. I want to highlight these people in the beginning because our team is actually a very diverse team combining cancer immunologists, cancer biologists, computational people like me, as well as bioinformaticians. And I'm very thankful to the previous speaker because our solutions are very much in line with the kind of solution that he came up with. Because of the lack of availability of any training data, we had to resort to prior knowledge incorporation. So that's what I will be highlighting predominantly. It's mostly all prior knowledge. So the key to our solution is actually to come up with a way by means of which we can have surrogates for TMB as well gene expression signature by means of which we can surrogate tumor mutation burden as well as PDL1. And what is the solution for that? It's ICR, which is the immunologic constant of rejection. What does ICR represent? It represents two opposite cancer immune phenotypes. Basically, the ICR signature consists of 20 genes, including Th1 interferon signaling genes, cytotoxic T cell effector genes, including the granzymes, CXCR chemokine ligands, as well as counter immune regulatory genes like CTLA4, PDL1 also. What they represent basically, if you have a high expression of these genes, it has been shown in the papers that are highlighted here that we can call them the tumors, we can call them immunologically hot. Basically, they have favorable response to immune therapy and they have favorable prognosis. Whereas when these genes are lowly expressed, then you have cold tumors with response to immune therapy as well as not favorable prognosis. So the ICR actually has been around for a long period of time for various different immune signature models. People have shown that across different cancer types, the ICR genes are actually part of immune checkpoint blockade based solutions. And in 2013, in an immunity paper, Jerome Gallon, as well as David Patagonepti, who is actually the brainchild of this entire challenge for us, they came up and identified these 20 genes based on the functional orientation of the tumors. And what we can ultimately say is basically across different immune signatures that have been associated with immune checkpoint blockade solutions, in prognostic, predictive, as well as in mechanistic models, the ICR genes consistently appear. This is pre-2013, but what we also did is post-2013, we collected many different data sets and we observed that several of the ICR genes are part of the solutions that basically are available. We analyzed several of these data sets which are highlighted in green, and ICR genes are constantly associated with a response to ICB. So our solution is actually, again, very simple. What we, in this keynote 22 trial organized by Merck, what they showed is that TMB can segregate out patients based on, has a prognostic value, and gene expression signature also has prognostic value when you are trying to segregate out patients who are going to respond and are not going to respond. And a combination of the two basically can help you to better segregate out these. So what was this GEP? The GEP is nothing but a gene expression pattern. And when we look at the gene expression pattern across the samples which are presented in this challenge, I mean, in this basically paper, when you correlate it with the ICR, it has greater than 99% correlation. So basically the T cell inflamed signature is nothing, it's very similar to the ICR signature. What I'm trying to come to the point is basically we can use ICR as one of those gene signatures that can help you to distinguish patients who are going to respond versus not going to respond. And at the same time, there will be no differences in the chemotherapy arm. Again, additional points which are trying to say here, again, in different data sets, when you have complete responders, you have partial responders and stable disease and progressive diseases. When you have responder versus non-responder, ICR across different cancer types was showing that you can basically differentiate responders from non-responders. So you can also see that the PDL1 was basically part of the ICR gene signature. So we have covered the PDL1 part, but now we want to find a surrogate for TMB. And what you observe is that the proliferation gene signature, a 5-set gene signature, is very well correlated in lung cancer, particularly with TMB. And TMB is actually, can differentiate responder versus non-responder in available immune checkpoint blockade data sets that are there, publicly available. So coming to our solution, what we do basically in the first challenge is we follow the outline of the challenge completely. Basically in the nivolumab arm, what they have said is that if you have high PDL1 and high TMB, then the patients tend to respond better and you have better survival. And then there are the other arms. So we basically came up with a simple rule-based framework which mimics exactly what was presented in the challenge. That if you have high TMB, in the patient profiles, if you have high TMB as well as high PDL1, and there are several patients for which the TMB scores and the PDL1 scores are missing, you can replace them with ICR high or with proliferation high and give these patients a score of 3, which is basically the higher score. And then similarly, we came up with simple rules to give scores of 2 and 1 to the other patients based on their TMB and PDL1 scores, as well as surrogates of proliferation and ICR scores. So here is a complicated formula to represent the same score that we have and how we decided like what is ICR high and what is ICR low. These are based on tertiles that you have, and we have the scores for all the patients. And we were the second solution, the second ranked solution in that particular challenge. In the overall survival challenge, again, we try to mimic the solution that was provided in the science paper in 2019, where you have TMB and GP high. So for equivalent, we use TMB high as well as PDL1 high or the ICR high group and gave them the score of 3. And then the others trying to mimic exactly what the solution was given in the science paper. We gave scores of 2 and 1. Again, the complicated mathematical formula, which is basically nothing but a linear additive model representing our solution. And we were the top performing team in this particular challenge. I'm very thankful to Mike also to highlight post-challenge also why our model works well is also because CTLA4 is part of the ICR gene signature. And in the keynote 227, checkmate 227, I think we are using a combination of nivolumab and pembrolizumab. Pembrolizumab basically, or ipilimumab. Ipilimumab is targeting CTLA4. And since CTLA4 is part of the ICR gene signature, our model tends to perform better even in that particular clinical trial. Finally, of course, we also tried to do some kind of machine learning by integrating all the publicly available datasets which are out there and trying to engineer scale invariant features, particularly like using single sample gene set enrichment analysis for cancer pathways, as well as the ICR and other scores that we try to include. But to our surprise and actually aligning with the previous speaker, ML models failed miserably. One reason is, of course, there is no training data that is available. So we are just throwing arrows in the dark. In the end, we submitted our trivial ICR model, which actually was ranked 7th in the sub-challenge. So finally, the take-home messages are that owing to lack of well-defined prior knowledge-driven model became paramount, and then simple linear model or a rule-based model had higher generalization capability as we see in the post-challenge result that we got to know. Finally, another important point is that the number of submissions made it very hard for us to optimize our model thoroughly. And I believe also that we can integrate information such as the TCR, DCR, as well as the Shannon entropy, other features better if we are allowed to give more tries. With that, I would also like to thank BMS and Sage for providing us the travel grant to come and present our results.